Hello, welcome back to Fresh Questions Update. Now we are on part 7 of Paper A Fresh Questions. This is a SPMM video crash class. Just reminding you that recent questions in the question bank get repeated in this exam. New questions generally appear from the same themes that are recently tested. So please go through these fresh questions as many times as you can. So question 1, according to Bronfenbrenner's model of contextual layers, which of the following can have a direct and immediate impact on an individual's development? A, peers at school. B, parental job loss. C, relationship between a child's family and the child's school. D, cultural norms. E, political system. So Bronfenbrenner described contextual layers of development. He described microsystem, mesosystem, exosystem, and macrosystem. This is a picture from Berk and Roberts' review of Bronfenbrenner's ecological systems theory. So what you see here is that the individual is surrounded by the microsystem, which is the immediate family. And then there is mesosystem, which is child care, the primary school, play area, etc. This is the mesosystem. And then you have exosystem, which is extended family, community health services, workplace, friends, neighbors, etc. And then it is surrounded by the macro system, which is the political makeup, values, etc. So according to this model, the more direct and immediate impact usually comes from the microsystem, the immediately surrounding system. As you can see, the microsystem is made up of immediate family, childcare, etc. And the connections among the microsystem is the mesosystem. So we can choose peers at school as the contextual layer which will have direct and immediate impact on an individual's development. So question 2, short tandem DNA repeat sequences of 2 to 5 base pairs throughout the genome is called copy number variation, chimera, microsatellite, mini satellite, macro satellite. So in your SPMM notes, you will see details of mini satellites, 2 to 5 nucleotide repeats. These are seen throughout the genome, 2 to 5 nucleotide repeats. The most common repeats are CA and GT. CA and GT are the most common. As you would have realized by this description, the trinucleotide repeats are a type of mini satellites. So there are 3 repeats, but it can vary between 2 and 5. The repeats are typically 5 to 50. They're hypermutable, they're unstable, so they can undergo mutation easily. Both micro and mini satellites belong to variable number of tandem repeats. So introns can be tandem repeats or interspersed, and the tandem repeats can be satellites, mini satellites, or micro satellites. The mini satellites are telomeric repeats or hypervariable repeats. So the answer for this question, 2 to 5 base pairs repeated throughout the genome, is mini satellite. Question 3. What is the process by which a DNA with the influence of RNA polymerase reads into a chain of RNA? Transition, transcription, translation, modification, synapses. So as you know, DNA reading into RNA is transcription. So DNA polymerase helps with replication, but RNA polymerase helps with transcription. And for translation, we have another enzyme, amino acid tRNA synthetase. So here, please choose transcription. That's the right answer. Question 4, which of the following receptors is on the somatodendritic part of the neuron? 5HT1A, 1D, 1C, 3, or 7? All of these are serotonergic receptors. Which of them is somatodendritic? So it's important to know that 5HT1A is an autoreceptor and it is present on the somatodendritic surface, because this is the mechanism by which SSRIs generally act. SSRIs primarily reduce the reuptake of serotonin in the somatodendritic area, the cell body area. Because of that, the serotonin availability in the immediate milieu of the dendrites increases. When that happens, the 5HT1A autoreceptors get superstimulated, so they will reduce the serotonergic output on a short-term basis. But over time, there's a downregulation of 5HT1A system happens, which in longer time actually helps the neuron to be more serotonergic in its output. It increases the serotonergic tone over time. That is why it takes a little while before SSRIs can increase serotonergic tone and produce relief from depression. So this is what is shown in this picture. Cell body is here. They have these little green things, which are 5-HT1A receptors. And when you take SSRIs, that's where the action usually starts. And the drama unfolds here before you see any synaptic action. So question number 4, the answer is 5-HT1A. Question 5, an autosomal recessive disorder shows Hardy-Weinberg equilibrium. It has an incidence of 1 in 6,400. The frequency of carriers of the disease is 1 in 32, 1 in 70, 1 in 80, 1 in 40, 1 in 64. Let's see another question as well. This is relevant. The next question, MCQ 6. Which of the following assumptions in a population is required for Hardy-Weinberg genetic equilibrium to exist? Random mating, presence of new onset polymorphic variations, presence of genetic drift, assortative mating, high rates of induced mutations. So what is this Hardy-Weinberger equilibrium? It keeps coming up in your exam. Hardy-Weinberger equilibrium. This is theory where the genetic makeup of a population is more or less standard and maintained if there is no great degree of mutations happening, if there is no non-random mating, if there is no mating, if there is no selective breeding, and if there is no selection drift or genetic drift. So provided these factors are not influencing the genetic constitution of a population, it's likely the genetic constitution will remain the same from generation to generation. So this is called Hardy-Weinberger equilibrium. The pool of genes remain constant, provided unusual influences do not exist. So if this is true, then mathematically we can actually predict how much will be the frequency of any given genotype, whether it's homozygote or heterozygote genotype. So if P is the frequency of the normal gene in the population, and if Q is the frequency of an abnormal gene, and if P, then P square becomes the frequency of normal homozygote, and Q square becomes the frequency of abnormal homozygote. Then 2PQ is the carrier frequency, and P plus Q will always be 1. This is the Hardy-Weinberger equilibrium. P plus Q remaining as 1 is the Hardy-Weinberger equilibrium. So for a given incidence, which is Q square, the frequency of the affected abnormal homozygote. So this is a recessive condition. So people are likely to be affected homozygotes, and the frequency is given as 1 in 6,400. So Q square here is 1 in 6,400. They're asking how many carriers are likely to be present, right? If you look at the question here, the frequency of carriers, right? So we know Q because Q square is 1 in 6,400. So Q is simply 1 by 80. Now they're asking for carrier frequency, which is 2PQ. Okay, so 2 into P and Q. So we'll assume P is small, and we'll just calculate 2Q. So 2Q is 2 multiplied by 1 by 80, and that will give you 1 by 40. So we're assuming P, normal homozygote, is small. We do not know that information, but we assume that. And generally this is okay. So we can calculate this as 1 by 40. So the answer is 1 by 40. And then they're asking what assumption is required for Hardy-Weinberg equilibrium to exist. The assumption is random mating. Mating should be random. Everybody should have equal probability of mating anyone in the population. If that is not present, if the mating is non-random, then the probability distribution will get skewed. Generally speaking, Hardy-Weinberg equilibrium exists because mating is reasonably random within the population. Question 7. Glycine and serine are co-transmitters at which of the following receptors? AMPA, GABA A, GABA B, endocannabinoid CB1, NMDA. So NMDA receptors are the receptors where glutamate binds. Glutamate binds to NMDA on the agonist site of the NMDA receptor. But the receptor also has many other allosteric sites. There's a site for glycine, a site for D-serine, D-cycloserine. There's a glutathione site as well. There's a polyamine site. There is a phencyclidine site on the inside of the receptor, on the gating side. There's also a ketamine site, diazosylpin site as well, diazoclipin site. There's a magnesium site as well. So there are many sites for NMDA receptor. Glycine and D-serine bind to the co-agonist site. This is called glycine modulatory site on the NMDA receptor. So for this question, the answer is NMDA receptor. That's where glycine and serine are co-transmitters. Which of the following can reduce groupthink? Seeking external opinion, pairing up group members, unplanned meetings of the group, strong chairperson, financial incentives. So what is groupthink? Groupthink, if you work as a group in a committee, group members succumb to conformity pressure, and they tend to agree with one another, and thus they pressure each other to avoid any necessary conflict. Conflict is necessary in a committee setting in order to arrive at a proper consensus, proper conclusion. But conflict would be avoided in many groups because of groupthink. Groupthink is the conformity pressure that makes people to agree with each other. So how can you reduce groupthink? Groupthink is not helpful all the time, but it makes the life easy. You can finish the meeting earlier, but it doesn't give you productive results. So how to reduce groupthink? Encourage open debate, acknowledge the presence of groupthink, seek external opinion, split the group into smaller discussion units, hold last chance meetings to encourage challenges, and finally, reserve the leader's opinion until everybody in the group has been heard. This is very important. So to reduce groupthink, you have to seek external opinion. Pairing group members, get crashing meetings, strong chairperson, giving money, these things do not help. Seeking external opinion will help. Question 9, which of the following compounds is used as a marker of GABA A receptor distribution in brain imaging? 11C flumazenil, 11C raclopride, PK11195, 18 fluorine 18 falipride, 18F septopuron. Which of them is GABA A distribution markers? Raclopride, as the name will tell you, it sounds like sulfuride, right? So it's a D2, D3 receptor marker. You also have falipride, it's also a D2, D3 receptor marker. Flumazenil, as you know, flumazenil is a benzodiazepine inverse agonist. So flumazenil is a GABA receptor agent. So you can use flumazenil in PET studies as in vivo marker of benzodiazepine receptor binding. Its iodinated analog called iomazenil that can also be used in SPECT. Flumazenil is an antagonist that binds with high affinity to benzodiazepine receptor. It's an antagonist. Okay, flumazenil is an antagonist for benzodiazepine receptors. So for this question, which of the following compounds is used as a marker for GABA A receptor distribution? The answer is 11C flumazenil. Question 10, which of the following is true with regard to institutional racism? Institutions are not capable of being racist. Individuals are fully responsible for their overtly racist behavior. Institutions cannot be held accountable. Institutional racism is less destructive as it is covered. Institutional racism can serve as a barrier to access health care. Institutional discrimination cannot occur without any awareness. The collective failure of an organization to provide an appropriate and professional service to people because of their color, culture, or ethnic origin is called institutional racism. It is seen in processes, attitudes, and behavior that amount to discrimination through unwitting prejudice, ignorance, thoughtlessness, and racist stereotyping, which disadvantages people in ethnic minority groups. It is as damaging as overt racism and discrimination. Which of the following is true with regard to institutional racism? The correct answer is institutional racism can serve as a barrier to access health care. Everything else is not correct. We cannot say institutions are not capable of being racist. They can have processes that are clearly racist. Individuals are fully responsible. The institution is not responsible is not correct. Institutions must be held accountable. Institutional racism is less destructive is not correct. It's as destructive as overt racism. Institutional discrimination cannot occur without any awareness is also incorrect. So that completes the part 7 of Fresh Question Update. Just to remind you again, practice from the question bank as much as you can. That's the only way to pass this exam. All the best.